Good evening. It really is an honour to be asked to give this lecture in memory of Don Ricks, and I read a little bit about him in preparation, and he obviously was a remarkable man, a doctor, an entrepreneur, and most of all, I think, a philanthropist. So it's all very well being 300, but actually it's much more fun being 10. And so I'd like to congratulate Genome British Columbia on its 10th birthday, and of course it's also the 10th birthday of Genome Canada. And indeed, it's the 10th anniversary of the publication of the draft sequence of the human genome. So what better moment for the birth of Genome British Columbia and of Genome Canada than the publication of that draft sequence of the human genome? And it's been an extraordinary 10 years since that draft was published, as I hope to show you in the next few minutes. So what I'm going to cover is genome sequencing itself, and then I'm going to give examples of how it's beginning to have an impact in obesity and diabetes, in cancer, and we had indeed a splendid visit to the Cancer Centre here yesterday, which has made some major contributions. I'm going to talk about infection, because I think medical microbiology is going to be transformed over the next few years by genomics. And I'm going to say a little bit about the future, because there are lots of medical implications of the human genome sequence, but there are also implications for society at large. So starting with genome sequencing. So the Human Genome Project was an extraordinary international consortium, and it really changed the way biomedical science was done. Early on, at a conference in Bermuda, it was agreed that all of the human sequence would be put into the public domain as it came off the sequencing machine. There would be no patenting of the sequences. It would just be put out there for anyone, anywhere in the world, to use as they wish. And that was a very important principle and has set the tone for all subsequent work in the genome area. So, what is the human genome? Well, it's 3.2 billion base pairs of DNA. People were a bit disappointed initially that there weren't more genes. We haven't really got any more genes than worms and flies, and indeed rather less than many plants and some fish. So roughly 20 to 25,000 genes. And only a relatively small fraction of that, less than 2%, actually codes for the proteins, which are the structural components of the body, or many of them. And initially it was thought that a large fraction, indeed large fraction probably are, junk. They've accumulated in our DNA attic, and we haven't had any way to throw them out. But we are discovering that more and more of the so-called junk may not be junk after all. It may actually have selective activity, it's been selected by evolution and have important biological activities. And so those small RNA species that Dermot talked about that were discovered by Fire and Meller and came from studies of how plants defend themselves against viruses are a good example of very important small DNA sequences in their coding that have only been discovered relatively recently. And really the genome you can take in two different directions. It's the blueprint for all of the proteins and the regulatory sequences, and you can do that to analyze how cells work, how proteins work, how genes work. The other route you can take is if you're interested in genetics, if you're interested in what it is that makes each one of us in this room different one from another, then studying the variation of the genome is of extraordinary interest. And I'll be focusing more on that than on the former. And of course we talk about the human genome, but it's also been fascinating to learn about the genomes of other species, so that we can see our place in nature, our relationship to other primates, the great apes, and indeed our close relationship in some way to those little worms that hang around in the soil, because it turns out that many of the most fundamental conserved proteins are very similar in both humans and worms. So, all very well knowing what the human genome is, but this is the problem, which is actually interpreting it. And I think interpreting the human genome is going to keep scientists busy for very many years to come. So, as I said, the first human genome was one thing, but it was actually understanding human variation. And the partnerships that were set up that started and sequenced the human genome have carried on and brought in industrial partnerships and And so this thing called the SNP consortium, which stands for single nucleotide polymorphism, those single base pair changes that differentiate each of us at millions of places in each of our genome, those were mapped as part of a public-private partnership involving public partners and many industrial partners who recognized that this was true pre-competitive research. It had very little intellectual property rights value in its own, and there was no point in duplication of effort. I'm not quite sure what's making this sort of, I'm probably spitting the microphone by accident. So the SNP consortium was the first thing that followed, and then something called HapMap, which started looking at the organization of each of these individual variants and how they're related one to another in different populations. And then having done that, it became possible to do human genetics on a scale that could never be conceived of before. So in the past, people would have taken maybe 50 people with disease X, and they would have compared samples from 50 or 100 healthy people, and they'd have looked at maybe genetic variants in one or two genes. But it rapidly became apparent that it was now possible to take everyone in this room and indeed multiply them by 5, all of whom had, say, diabetes, and compare 500,000 genetic variants with the same number of variants in a very large healthy population. And that ramped up the power of genetics to discover variation in both health and disease. And so there's a list there of examples of variation associated with both infectious and acquired disease. So the explosion of DNA sequencing is quite extraordinary. And what this graph shows is from one genome centre, and that's the Sanger Institute. We fund it, the Wellcome Trust funds it, it's just outside Cambridge. At the Sanger Institute, John Soston and his colleagues sequenced a third of the human genome. If you look at the line very near the bottom, that was the total sequence that had been generated everywhere in the world on the European Molecular Biology Organization computers to 2007. And the extraordinary graph shows the cumulated DNA sequence that's been generated at the Sanger Institute since 2008. And you can see that it completely dwarfs everything that had been generated up until 2007. And the ticker tape at the bottom, the TAT, GCC, etc., when you went into the Sanger Institute, you used to see these letters flying across a display like that. That's had to be switched off because it can't really cope with 1 million bases a second. I mean, you can't just see that anymore. So there's been this extraordinary technological advance. And to put this in some kind of financial context, the first human genome took about 10 years to sequence. It cost about $1 billion, and it involved scientists from all over the world. By 2009, there had been 200 human genomes sequenced, and a human genome sequenced using one of the platforms probably cost around $50,000. The price now has come to around $10,000 a genome. And in 2011, literally thousands of genomes will be sequenced for research purposes around the world. And then I think it's a projection, but I think that everyone believes it's quite likely that by 2015, it'll be possible to get a complete human genome for less than $1,000. And of course, that then opens it up to many people in this room who might, out of curiosity, and I would say that by 2015 it will still probably be for personal curiosity, I may be wrong. It really will become possible to do genomes on a patient-by-patient, person-by-person basis. This graph shows the discovery of genes associated with complex diseases. These aren't the so-called monogenic disorders such as cystic fibrosis or Huntington's chorea, where a mutation in a single gene is sufficient to cause quite a serious illness. These are the common diseases: diabetes, hypertension, many forms of cancer, age-related macular degeneration. You can read the list. And it proved incredibly difficult. We knew there was a strong genetic component to these diseases, but actually identifying the genes proved incredibly difficult. So 2000 was a triumph. PPAR gamma, a single gene, was found to be associated with cardiovascular disease. 2001 was wonderful. 2 genes were discovered that year, both associated with inflammatory bowel disease. But now look to 2008 and 2009, and you can see that in 2009 it's no longer possible to write down the individual loci on one slide, because well over 500 different variants were found associated with disease. These are extraordinary discoveries. Now, and this was the front page of one of the UK broadsheets, The Independent, when the results of a study that we'd funded called the Wellcome Trust Case Control Consortium was first published, showing a whole range of different diseases in which genes were starting to be found. Although some of them have proved much harder than others, so hypertension has proved very difficult to crack, as indeed has bipolar disorder. Some people have been disappointed. They've said, well, each of these genes only carries a relatively small increased risk, and you're not going to be able to predict your future from, or indeed whether someone's likely to get a disease from discovery of these genes. And indeed, that is correct. But as I'll show you in a minute, what these discoveries are generating is new hypotheses about the cause of these common diseases, new pathways, and that in turn will lead to new paths of drug discovery and other interventions. So that's why this is so important. Now, moving on, a project that's going on at the moment, and in fact just a couple of weeks ago, the first results from the 1000 Genome Project were published. So we had the Human Genome Project, we've now got the 1000 Human Genome Project. Again, it's a global collaboration, North American partners, UK, China, and again, the information is being put out into the public domain in real time. And this just shows some of the populations that are being studied from around the world. So the 1000 Genome Project will expand our knowledge of human genetic variation, and it will expand our knowledge about how humans migrated around the planet. And already this information is giving extraordinarily detailed information about the relation of one population to another. Indeed, if you look at populations in West Africa that speak different languages, then in fact you can find that there are genetic variations which associate. They don't cause the different languages to be spoken, but they reflect the fact that the population separated at different times, and so you can identify genetic heterogeneity according to whether someone speaks Wolof or another language in the Gambia. Now, this is what it means for each of us. So each of us has 3 or 4 million distinct variants in our genome, which will separate each one of us from other people in the room. 10 to 12,000 of these are so-called non-synonymous changes. In other words, they will change an amino acid sequence in a protein. And that may or may not have a functional consequence. There's 200 to 250 that will actually either have an increased number of amino acids or a decreased number. 70 to 100 genes which will actually be knocked out functionally because they'll have premature signals in them that tell them to stop being transcribed. 20 to 50 will have quite major disruptions, and every one of us is likely to have lurking in our genomes somewhere between 50 and 100 what are known as recessive disease-causing mutations, and are already in the Human Genome Mutation Database. And that, of course, is why it's a relatively risky business to marry your first cousin. Because it's when you marry your first cousin that the two, and these are mainly recessive, in other words, you have to have 2 of these alleles meeting each other, one from mother, one from father. When you marry your first cousin, the risk of that happening is much higher. And so in populations where intrafamilial marriage is relatively common, then you do see more genetic defects, and that's been known for generations. So none of us should be too prissy about our genomes. We've all got lots wrong with them. And this is a project that we funded last year. It's called the 10,000, it's known as the UK 10K project, and that will be complete by 2013. And this aims to sequence fully many patients who've got neurodevelopmental diseases, severe obesity, rare disease traits, to really dig into the genetic causes of significant diseases. So this has been the most extraordinary journey in a very short time. So let's take a few specific examples. Now, one thing that I think is extremely important to get across is that We are a complicated and messy mixture of our genes and our